Welcome students for the new topic that we are going to discuss today is reproduction in fungi. So reproduction is a phenomena where a particular organism develop its own organisms. So in fungi, reproduction mostly takes place by vegetative, asexual and sexual means. During asexual and sexual reproduction process, spores are the essential structures. Spores can be formed after meiosis called the miospores, as in case of ascospores, basidiospores and sporangiospores, and those resulting from mitosis are called mitospores, which are zoospores, eplanospores, conidiospores and eurydiaspores. The diploid body produced as a result of sexual fusion is known as zygote. In lower fungi, it is termed as resting spores, whose spores are zygospores, while in higher fungi, the zygote is represented by a diploid nucleus produced in the fruiting structures called ascus or basidia. The diploid nucleus, after undergoing meiosis, form haploid nuclei serving for the haploid sexual spores called ascospores and the basidiospores. So vegetative reproduction is the reproduction, where said it is of two types, vegetative and cellular. So there are different methods. So vegetative reproduction, mostly under favorable conditions, it will develop, and this vegetative reproduction in fungi takes place by means of fragmentation, fission, budding, odia, chlamydospores, rhizomorphs and sclerotia. So fragmentation is a result from accidental severing of the mycelium into beds of fragments by mechanical injuries, and the mycelium breaks into segment of hyphae. Each segment further divides of the cells, apical growth develops and a new mycelium will be developing under favorable condition. Fission is a condition where the single cell multiplies by fission. In unicellular fungi, like fission is the single cell multiplies by fission. Parent cell elongates and divides. Here the cell elongates and divides transversely into two daughter cells. First the nucleus divide, followed by the cytoplasmic division, and the two daughter cells separate and leads their independent life. This phenomena we call it as the fission. The third type of vegetative reproduction is called budding. This is common in the yeast, where the cell reproduced by a process called budding. The parent cell puts up a small outgrowth which is called the bud. Bud enlarges and separates from the parent cell by a cross wall and develops into a new individual yeast cell. And this separation from the parent cell produces a new bud, and in this way the chain of buds may be produced and get separated. So this is the phenomena of budding taking place in the cell. So this is the parental cell, the outgrowth will develop, and that will got, and in this way many buds will develop, and the bud will separate as independent and will develop separates from the mother cell and will act as an independent. The other process of vegetative reproduction, we call it as odia. In this, some of the filamentous hyphae break up into individual cells, which we call it as the oidia or orthospore, and each orthospore develop into a new mycelium. Here the odia becomes rounded, oval in shape and appears like a beads of a rosary. The other type of the reproduction, asexual reproduction or vegetative reproduction, we call it as the chlamydospores. These are one cell fragments which function as perinating bodies formed either singly or in chains in the vegetative hyphae. These chlamydospores develop a thick resistance wall and accumulate food material and help the fungus to come over the unfavorable conditions. With the return of the favorable condition, each chlamydospores develop into a new mycelium which is found in fusarium and euchre. So these are the chlamydospore formation, which forms a thick wall around it. The other type is called reproduction. We call it as the rhizomorphs, where several hyphae interwoven and form a rope-like structures. And this rope-like structures we call it as the rhizomorphs. Under favorable conditions, they resume the growth and give rise to new mycelia. They are root-like strands, usually dark brown in color. They are dark brown in color. Under favorable conditions, these remain dormant, but with the onset of the favorable conditions, these rhizomas resume growth and may give rise to the fruiting bodies. So these are the root-like structures that are being formed, which we call it as the rhizomorphs. The other we call it as the sclerotia. These are the perinatic bodies formed by the compact mass of interwoven hyphae. These, under suitable conditions, germinate and form a new individual called claviceps and sclerotina. Asexual reproduction in fungi is the most common method. It is repeated several times in the season. It takes place by formation of the spores. The formations of the spores is called sporulation. Each spore develops into a new mycelia, and these spores which are being formed by as a result of mitosis are called as the mitospores. So these spores, they vary in their color, shape and size and arrangement of the hyphae. They may be hyaline, green, yellow, orange, red, brown to black in color. They may be minute to large in size. They are globose to oval, needle-shaped to helical. They may be uninucleate or multinucleate. In some fungi like Alternaria and Curvularia, they are multicellular. So the mitospores or the asexual spores, when you see, there are of three types. One is known as conidiospores, the zoospores and the sporangiospores. The conidiospores, spores produced by the conidiospores. The zoospores are the motile spores with flagella for the movement, and the other is the sporangiospores formed within the sporangium. So this is the sporangiospore. It is an example of rhizopus with the sporangiospores. They are aplanospores. They are non-motile and lack flagella, formed inside the sporangium, and they may be uninucleate to multinucleate and possess two layers of the cell wall. Then the second type of the asexual reproduction is zoospores. It is found in Pythium, lower fungi Pythium. They are naked spores which are swarming, encysted and secrete a cell wall and germinate by germ tube into the thallus. They are equipped by one or two flagella. The third type of asexual reproduction is called conidiospores. In this, the conidia are non-motile, deciduous mitospores formed extremely as a single separate cell. They develop directly on the mycelium or on different hyphae called conidiospores. These conidia are produced singly in case of Phytophthora or in chains in the tips of the conidiospores in case of Aspergillus, and as at the tips of the branches as Penicillium in case of Penicillium. So conidiospores, they are allied singly or scattered in the mycelium. According to they are termed as Simnima, Sporodacea, Eservioli, Pycnidia, flower-shaped, or pustules. The sexual reproduction in fungi is called the perfect stage. It involves the fusion of two compatible sexes called the gametes or opposite strains. The fungal sex organs are gametangia. They may be equal in size. In higher ascospores, they are morphologically different. Different gametangia are formed. The male gametangia is called antheridia, the female the escogonia. The fungus may be homothallic, that is, the gametes come from the same mycelium, or heterothallic, fusing of the gametes come from the different strains of the mycelium. So the so these are the reproductive process that takes place. One is the plasmogamy, where the fusion of the cytoplasm of the two parent cells, the karyogamy, the fusion of the two nuclei from the parent cells, and the meiosis, where the cell division that reduces the chromosome number. So this is the sexual reproduction which takes place by plasmogamy, the karyogamy, and meiosis. So plasmogamy is the union of the protoplast of the reproductive hyphae or the cells. So in this, the two nuclei do not fuse with each other and they are called dikaryons. So this is the unique to fungi which continues for several generations and divides simultaneously during cell division. They are passed to the daughter hyphae. Karyogamy is the fusion of the two nuclei which takes place in the next phase called karyogamy. It immediately follow plasmogamy in case of lower fungi or may take a delayed long time in higher fungi. Karyogamy eventually occurs in all sexually reproduction fungi, sooner or later followed by meiosis producing 4 genetically different spores. So these are the different methods of the plasmogamy. They are the somatogamy, spermatization, gametangial copulation, gametangial contact, and planogametic copulation. In somatogamy, it involves the fusion of the somatic cells to create a new form. In spermatization, a method where sperm cells perinate a receptive structure, and in gametangial copulation involves the direct fusion of the gametangia to form the new cells, and in gametangial contact the process where the gametangia come into close proximity and only the nuclei of one gametangia will enter into the other. And planogametic application involves the motile gametes merging to form a zygote. So in the planogametic copulation, there are three types. One is known as isogamy, anisogamy, and oogamy. In isogamy, both the gametes are similar. Anisogamy is found in allomyces and chytrid. Oogamy, in this one is motile and the other is non-motile. Oogamy is seen in fungi of like Pythium and Albuco. The other type is the gametangial contact, where the oogonia and the antheridium come in contact through a tube and one or more nuclei. They migrate into the oogonium and they as observed in the penicillium. This is the gametangial copulation. This is found in mucor and rhizobus, where the gametangia make contact and the entire content of the two fuse together. Somatogamy means the two cells are similar. In higher fungi, ascospores, basidiospores, there is progressive degeneration in sexuality. The entire process is very much simplified by fusing of the two mycelia which belongs to opposite the strains. The post-fertilization stage results in production of the fruiting bodies called ascocarp and the basidiocarp. Heterothallism was first used by American geneticist called Blacksley in 1904. It is a condition of sexual reproduction in certain species of order Mucorales, where they observed that some species of the rhizopus, when two mycelia of different states were allowed to come in contact with each other, they developed the zygospores. Heterothallism is defined as a condition in which zygospore formation takes place only when mycelia arising from asexual spores of two genetically different mating types, plus and minus, are allowed to interact. So this heterothallism, so there is a difference between homothallism and heterothallism. Homothallic species require mycelia of only one strain to interact and formation of zygospore. Mycelium here is bisexual, while the heterothallism species require two different strains to interact to enable the zygospores to be formed. This mycelium is unisexual. Plus and minus strains represent the two different sexes. So these are the different spores that are being, so this is the zygospore formation indicating the heterothallism, plus and the minus strains coming together. So there are different types of heterothallisms that have been observed. One is based on the morphology, we call it as the morphological heterothallism, and the other, which though externally it looks similar but internally it differs, we call it as the physiological heterothallism. It is 2-allele heterothallism and multiple allele heterothallism. Morphological heterothallism is the condition when morphologically different sex organs are produced or two closely associated mycelia. The sex organs are morphologically different, as is found in Acalia ambisexualis and blastocola. Physiological heterothallism, these are two morphologically similar but physiologically different hyphae, and it is found on two allele heterothallism, where there is a one as a allele is plus and the other allele is the minus. So here their genetically different characters are represented by A and A located on the same locus of the allele of the chromosome. So this mycelia of plus and minus are self-sterile or self-incompatible, and this is found in Neurospora, Mucor, and Puccinia graminis. Multiple heterothallism means that more than two alleles determine the sexual compatibility located at one or two loci. They may be bipolar multiple allele heterothallism or tetrapolar multiple thalli heterothallism. So bipolar means that the heterothallism is controlled by multiple alleles at a single locus, and tetrapolar it is controlled by multiple alleles at two loci. There is another condition of reproduction that is known as the parasexuality, which was discovered in 1952 by Pontecorvo and Roper at the University of Glasgow in Aspergillus nidulans, or the imperfect stage of Emericilla nidulans. So parasexual cycle occurs in those fungi in which true sexual cycle does not take place. Parasexuality is a cycle in which plasmogamy, karyogamy and meiosis takes place in a sequence but not at a specific time or at a specific point in the life cycle of the organism. This parasexual cycle is also known as somatic recombination, and this has been identified in several imperfect fungi like basidiomycetes and ascomycetes. So this is Pontecorvo's 1958 idea of the parasexuality. So you can see in the this is the strain 2. This strain 2 is step 1. It will reduces the nuclei. The heterokaryon is produced. The diploid conidia is there. It will again develop into a new mycelium. So this, you can see the haplodization process of the parasexuality. So what are the steps involved in the parasexual cycle? So steps involved in the heterokaryotic mycelium formation. First is the haplodization, followed by diploid sorting, mitotic crossing over, diploid multiplication, nuclear formation, nuclear fusion will take place. So these are the different steps that are involved in the formation of the heterokaryotic mycelium. So if you see the comparison between sexual and parasexual cycle, in sexual cycle nuclear fusion is specialized structure, in parasexual no nuclear fusion in vegetative structure. Zygote persists one nuclear generation, while in parasexual it persists through many mitoses. Here recombination by meiosis, crossing over and reduction of chromosome number. Here recombination takes place by rare mitosis or mitosis crossing over at random assortment of the chromosomes. In sexual cycle, products readily recognize and isolate. In the parasexual, recognized only by suitable markers. So the questions related to this particular topic are: what do you understand by heterothallism? Highlight the pattern of distribution of sex in fungi. Discuss the parasexuality in fungi. So these are the references from where the PPT has been created, and any queries or you can look inquire with the concerned teacher.